question asked, let's see if I remember this right. It asked you to track a carbon through the citric acid cycle, and the carbon it asked you to track was that one from acetyl-CoA?
No,
It was the middle one? Dang it. Fine, whatever. Okay, the one with the carbon there. Okay, so I would argue in that first step that we could track it down relatively easily, but it actually starts to get very dicey after that. So it is going to be one of those carbonyl carbons. Now, which one becomes kind of tricky, particularly as you then move through the rest of this reaction. Which one is that carbonyl? Well, the molecule is symmetric to begin with, so how do you know where that carbon goes? That has to do with how the enzyme grabs the molecule, okay? Because we are forming a chiral atom. So there are, each half of that molecule has a name to it, and I believe they actually call it R, and I'm going to guess L for the right-hand side and the L side, of the left-hand side of the molecule. The enzyme grabs at that point and prevents the chemistry from occurring there. Okay? So you end up seeing the carbon move through the structure in kind of an odd fashion that you would have to know based off of where the acetyl-CoA came in. Okay? So immediately that makes that very, very tricky in that very next step. What makes it even worse is, let's say that we do end up tracking it, and I believe it's actually out here that it keeps it. What happens now? Where did it go? So if we're now here, which carbon?
It flips, doesn't it?
It rotates.
We lose track of it.
Yeah.
It could be either of those two positions. So in the question, you'd have to label both, because both of them are equally likely to have it.
That's what it meant when it said that.
Yeah. Then it continues to track through as both, pretty much all the way through the rest of that. Whoops.
Oh.
Yeah, I did that right. Okay. And that's because we've just lost track of it. And once you spit that molecule out, we have no idea where the radio label is because they're both the same.
Right.
Okay. So when I saw the question, I was like, that's like the glycolysis one. I like that one. We want to keep that because we want to see how it tracks through. Went way difficult, way fast. So I removed that question because we definitely didn't go into it in that level of detail. Okay. So I'll put it, try and get it into the practice. So if you're interested in seeing what happens with that carbon and thinking about how it translates through, the explanation for the question actually does a pretty good job of explaining what's happening right here at the beginning part on how the enzyme actually grabs and effectively protects the carbon from your acetyl-CoA from doing any of the chemistry. Okay, so kind of a neat question, just unfortunately more detail than we needed for that. We've already seen this slide. This is kind of our overall summary. Actually, before we do that, we kind of okay with 19. We got one slide left in 19, and that's it. And it's a really simple slide. Okay. Then we're done with chapter 19, and we're moving into chapter 20, which is a really kind of odd chapter in my opinion. I felt that the text itself was kind of confusing and went in a lot more detail than you really needed. You need to pick up some kind of basics out of chapter 20, and with those basics, you can answer all the questions in the homework. It does take some reasoning and kind of comparing through things, but I think the questions in chapter 20 with some kind of basic understanding of the electron transport chain is actually pretty straightforward. Okay. Still, not saying it's easy, but you don't need a lot of the detail that the chapter throws at you. Okay. So what we've gone through and done is we did glycolysis, which took us to pyruvate. Once we've got the pyruvate, we move into our, through that big enzyme into our citric acid cycle. And what we're doing in both those cases is ultimately trying to produce energy. Okay, so we got a little bit of ATP out of glycolysis, we got the equivalent of ATP out of the citric acid cycle with GTP. But ultimately, those aren't big energy drivers out of this. What we're really producing in both those cases is NADH. We're producing electron carriers. Those electron carriers are really where our big energy production happens, and that's in the electron transport chain. So with both these mechanisms, cycles that we're looking at, what we're trying to produce is that high energy electron that we can then dump into oxygen, turn it into water. Okay? So it's that oxidative energy production that we're looking at. And that's our electron transport chain, which is really happening down at this end part. Okay? So what they're trying to show with this figure is that everything is goal is to make your NADH and FADH and ultimately a couple other pieces that we'll talk about as our electron carriers. Once we've got the electron carriers, we can then dump those electrons into that last step, which is our oxidative process, and ultimately oxidative phosphorylation, because we're producing ATP, a lot of ATP out of that last sequence. Okay. Oh, there's a nice little figure. For some reason that did not show up in my slides, so it's a little bit of a surprise to me. What we're looking at here is actually your mitochondria, or kind of a zoom in of your mitochondria. We'll get another picture of this. This is your outer membrane of your mitochondria. We have an inner membrane of the mitochondria. Your citric acid cycle is happening on that very inside of this. And then your oxidative phosphorylation as a chain is happening through there. So that's our oxidative. It's just sort of oxidative P. This was our outer membrane and our inner membrane. This is just giving us an idea of where everything is occurring. Glycolysis is actually happening out in our cytosol, which is a whole other issue that we can address at the tail end of this chapter. Okay? So we're trying to kind of get everything into the mitochondria because that's where all of our energy is coming from. Okay. So features of our cycles that we looked at in our glycolysis and our citric acid cycle. So I took out some of the pictures that we've got from our previous slides and then tried to go through and summarize these. Within our glycolysis, we end up producing 2 ATP. But that says 4 ATP. Why is it only 2? We consumed 2 in that process. So we're looking at the net, the overall. We end up getting 2 NADH out of that process, and we get 2 pyruvates. Okay. The 2 pyruvates become important, and this is something that shows up in the Chapter 19 homework, right, is that we are running the citric acid cycle twice. Why do we run it twice? We're only putting in a single pyruvate per cycle. Glycolysis gives us 2. Okay, so for a single glucose molecule, we end up producing 2 pyruvates, which means when we move to the citric acid cycle, okay, we have to look at running that reaction or that cycle twice. So looking at those totals that come out of that. I believe I totaled that correctly. We got the 2 NADH. We've got, and I put ATP in quotes. Why did I put ATP in quotes? It's not officially ATP is actually why I did that. It's GTP, not ATP, right, which is happening when we convert our succinyl-CoA into our succinate. Okay. It's still a high-energy molecule. It's just it's not our main currency for transferring that phosphate around. Okay. So it's still an energy source. It's just not the same. And then the last one that we tagged or that I added on here that we haven't talked about yet is coenzyme Q. Okay. That's going to be important because that's happening right in here. So we were talking about our transfer from succinate to fumarate. Remember how that was kind of a weird process because we did, oh man, those structures don't look very nice. Let's see if I can erase those so we can see the structure a little bit cleaner on those. Okay, our fumarate, I think I've got my carbons right, was that alkene, and our succinyl-CoA was the alkane. Okay. So what reaction did we do to go from our succinate to our fumarate? Can everybody see those? Should I try and clean that up a little bit more?
From the
looking at bottom from the left.
We're looking at the bottom, so we're going from our succinate to our fumarate.
What you're doing is breaking a pi bond.
It's not breaking a pi bond, we're creating a pi bond.
Oh, I'm sorry. I went top to bottom.
Bottom to top, bottom to top, remember our circle around.
Yeah.
Okay. So it's an elimination reaction, okay? But it's not a simple elimination reaction that we've done before where we eliminated water. This was a weird one, because what are we eliminating from the molecule? 2 hydrogens, okay? Which is not an easy process to do. And this was one of the things we talked about, well, what thing would allow us to do that transfer? NADH was a possibility, but NADH dealt with polar hydrides. We don't have a polar hydride here. And so what we said was being used was FAD. What's the problem with your FAD? Okay. There were 2 things when we talked about our electron carriers way, way, way back when. NAD and our FAD. Okay. NAD is small, FAD is massive. What's that going to be an issue with? Sort of.
But it's size, right?
It has to do with our size, which is the equivalent of steric.
Stability of the.
Kinda, sorta. Rotations bring it in too. We're trying to get things moving from one site to another site. The bigger it is, the more difficult it is to move. Okay? So it turns out our FAD tends to be an electron carrier, but it's very immobile. It stays pretty much locked within the enzyme. So while it does temporarily transfer those electrons nicely, and we can argue all day about how it does that mechanistically, okay, because it very likely, as Diane was pointing out, is potentially a radical mechanism because those are very nonpolar bonds. Okay? So if it doesn't move, how can we get those electrons then to our electron transport chain? Okay? Well, it has to go through a cofactor. That cofactor happens to be coenzyme Q. Okay? So when we look at this version of it, you'll notice they aren't showing FAD because FAD is, while it's part of the enzyme and it is what is actually accepting the electrons, it can't go anywhere. So it ends up dumping those electrons off onto CoQ, okay, or coenzyme Q. Coenzyme Q can then transport those. Okay? And that step is actually a very important step that shows up kind of oddly in the electron transport chain, which is very weird because we're saying it's part of the citric acid cycle. Okay? So it kind of parallels in both. Okay? And we'll talk about that when we get into our electron transport chain. But ultimately, what we're trying to see here is that we've got these big summaries. We're starting to get some ATP, some energy out of this. But what we now need to do is convert our electron carriers, get those to really help us produce more energy. And to do that, we're still trying to form ATP. But the problem with forming ATP is not a redox reaction. It's just the transfer of a phosphate. So how do we couple, okay, redox reactions to phosphorylation reactions? Okay, so that's what's happening within our electron transport chain, is a coupling of those 2 aspects. So we can shut down that coupling and still allow for ATP synthesis to occur on its own without that coupling, or we can destroy the electron transport chain or destroy our ATP reaction but still generate our electron transport chain or still have that functioning. Okay, so these are 2 independent things that the cells have designed to react with each other. Okay, so they end up facilitating one kind of transfer versus the other. Okay, so this slide is ultimately the same thing as the last one. All we're trying to do with the citric acid cycle and our glycolysis is end up generating these electron carriers, our NADH and our FADH2, which are really trying to transfer those electrons through. Okay, so that gets us to chapter 20. We got a lot of different things happening within this. This is pretty much the text of what's happening. Whoops. Oh, man. I totally switched up the sequence of my slides. Oh, well. This is the text of what's kind of pictorially going to be shown now, probably like 20 slides later. Okay. But what we're looking at is the connection of multiple different subunits facilitating the transfer of electrons, okay, from a high energy state to a relatively low energy state. So we're taking free electrons, ultimately stored in a hydride, and we're dumping them onto oxygen. Okay, well, why would that be a good thing? Oxygen is more electronegative than hydrides. That's going to be a more stable process. Okay, so what we're trying to do is kind of that exothermic transition, high energy to low energy. But if we do it all at once, like for instance throwing a match into your gas cylinder or your gas tank in your car, that could be a bit of a problem. So we want to stepwise down that energy process. Okay? And that's a lot of what the electron transport chain is doing, is facilitating that and slowly dropping the energy of the electrons so that we don't get this massive release of energy that's uncontrollable. Okay? We're trying to control how it goes through and does it. Okay? All of this is happening within your mitochondria. Okay? And the biggest thing that's happening within this is that we got this whole citric acid cycle stuff. Well, that's an eraser. We've got our citric acid cycle happening in the middle there, generating all of these high energy electron carriers. Those electron carriers then interact with the electron transport chain, which is now sitting on, if I can do this, on that inner membrane of the mitochondria. Right? And what those end up doing is transferring hydrogens across that membrane, okay, into the intermembrane space. Okay? So the space between the membranes, because we've got an outer membrane and an inner membrane. So what ends up happening is on one side of that membrane, we transfer those hydrogens out into that intermembrane space, we have a high concentration of H+. Okay? Which we could then say is acidic, so we can bring in pH, high, lows, all that fun stuff. Okay? Which means also, necessarily, because we're transferring hydrogens out, that what's happening on the inside? It's going to be a low concentration of H+. Okay? So what we have is on one side of the membrane, a lot of positive charge. And on the other side of the membrane, we have a complete lacking of positive charge, which would mean negative charge. Where else have we seen positives and negatives? In your batteries. Okay, what we're generating is an electric potential, okay, a very high energy state between those two charges. The natural system is going to want to do what? Stabilize it by evening that charge out. Okay? So that brings up a first issue. Most membranes tend to be very permeable. They allow the passage of at least small ions through them. This inner membrane, not very permeable, particularly to H+. Because the whole point of this membrane is to establish that gradient. Okay? So if that membrane allows for the free passage of H+ through it, we can't control how the energy transfers through. Okay? A nice analogy could be looking at a dam. Okay? Why do we dam up rivers? Well, there's lots of reasons, but what's one of our big reasons?
To produce energy.
To produce energy. Water is flowing from a naturally high state at our high altitudes, flowing down towards our oceans, which are lower altitudes. That's energy released as it moves from a high potential to a low potential. Okay, so what we do with dams is put something in the way of it, and now we say, okay, now I'm controlling how that energy is released. And if I can use that energy to pass through my own generation system, my electrical energy system, I can now produce energy by its natural flow down. That's what's happening here. It's the same thing. We generate a high H+ concentration that naturally wants to flow back across. Okay? But what the mitochondria has done has built a dam to prevent that natural flow, that natural neutralization. Okay? We then need something to facilitate the transfer across and use that as our energy carrier. Okay? So we have a couple big things coming out of this. Our electron transport chain builds that gradient, and then we take advantage of that gradient to produce energy. Okay? Another term that might float up here, Christi. Anybody know how to say that? Christi. Okay. What is a Christi? Okay, good. You guys know more than I do. That's good. I like that. that's looking at the folds within your inner membrane. Okay, so it's not the inner membrane itself, it's the little folds and bubbles within it. Okay.
Which increases the surface area.
Which is going to increase the surface area of that inner membrane, which could potentially then increase the likelihood of putting more things in there to generate more power. So, this whole electron transport process is exceptionally complex, and there's a variety of different ways that we can look at it. So one version is to kind of look at this attempt to color things, and your textbook has done kind of a neat job on this. What we are doing within this is we're taking our high energy molecules like NADH and our FADH2, and we are transferring those, I did just circle those, but our NADH and our FADH2, which in theory will show up circled on our screen here in a second, right? And we're transferring those electrons into the electron transport chain, and that's what's happening through the middle of this. Right? And what they're trying to show is all of the kind of important molecules involved in accepting those electrons and doing those shifts through it. They've also gone through and tried to color code these according to the individual sections of what's happening within them. Okay? So if I remember this right, what we've got is your first complex. You've got, this is your second complex. This is your third complex. This is your fourth. Did those actually show? Those did show. Right. Why did I not circle everything? Well, what's happening with those other systems is those are actually the transfer agents to move from one complex to the next complex. So an interesting question that you might ask is, well, don't we already have a transfer agent for those electrons? Good question. I don't have an answer for that. Okay. So those coenzyme Q and cytochrome C are your electron transport agents within the electron transport chain. Coenzyme Q is membrane bound. It's running through the middle of the membrane. Okay? So we could see a legitimate reason why we don't use NADH versus coenzyme Q, because NADH is relatively polar. Would that make it into the membrane? No. Okay. Cytochrome C is kind of an interesting one on its own. Not membrane bound, which we'll see in one of the pictures. It actually floats over the top of the membrane. Okay, so why do we need cytochrome C? Okay, well, that's another good question. So I kind of got interested enough in trying to put the slides together real quickly and looked at some of the other processes of it. It is very tightly associated with the membrane, okay, but it is not officially membrane bound. One of the interesting processes is that cytochrome C, if it makes it outside of the mitochondrial electron transport chain, contributes to cell death. So it is kind of tied to the electron transport chain, and if all of a sudden that falls apart, well, you're stopped producing energy. Very likely we want the cell to die so the cytochrome C can come off and is associated with apoptosis or cell death. Is there a question there, Diane?
No, I was just was just checking, because in the book it actually describes the complex one is going from the NADH transmitted all the way to the CoQ.
Yes. So that's what's happening here. The NADH comes in.
Right.
That's complex one accepting those electrons. Complex one shuttles those electrons to CoQ. CoQ then shuttles those electrons to complex 3. Okay. So CoQ is kind of in a weird state because there's two things that feed into it, complex 1 and complex 2.
See, and that's where I think it gets a little confusing
Yeah.
when you're reading it, because it describes it and it breaks down that first reaction, but then it carries on the second reaction from the electrons actually ending in the CoQ, and then the second one is picking back up.
Yeah.
So that's right. Thank you.
What is complex 2? Where did FADH2 get produced? In the citric acid cycle. Okay. Complex 2 is actually part of our citric acid cycle. It's a membrane-bound protein because what it's doing is facilitating electron transfer in our electron transport chain, which is a question, I think, in Chapter 19. The enzyme that facilitates that from succinate to fumarate, okay, that is a membrane-bound protein. it is producing FADH2. But FADH2, we said, was relatively tightly bound within the enzyme. That's not going to be an easy way to transport electrons. So what does it do? Because it's in the membrane, it can transport those electrons to CoQ. CoQ can now transport electrons further into our electron transport chain.
Point of clarification, you are getting electrons from both the NADH and the FADH2.
Yes. Both of them produce electrons. Both of those electrons will cycle into CoQ and come through the electron transport chain. Not simultaneously.
So 1 and 2 are both transferring electrons to CoQ.
1 and 2 both transfer electrons, and CoQ is our acceptor of those electrons. It can only accept one set at a time. So when we're looking at that membrane, it's not a single one of each of these. You're looking at probably thousands of these, if not millions, of these complexes within an individual system.
And it's coming from the succinate.
Yes, complex 2 is our succinate to fumarate. Okay.
What is the third one do? It bypasses?
We'll get to it.
coenzyme Q?
No, coenzyme Q shuttles electrons to the third complex. Okay, so we're doing another electron transfer mechanism. Our transporter is CoQ to complex 3. Complex 3 shuttles those electrons to another transferer, cytochrome C. Cytochrome C ultimately transfers those to complex 4, which facilitates the final reduction of, oxidation of oxygen gas into water.
So CoQ is going through a series of redox.
Yes.
Reactions from-
So that's going to be one of the fun things with this, which the textbook goes into a lot of detail, and I cut all of those, most of those homework questions out. All of this is redox. Okay. You are transferring electrons from one molecule to the next to the next. All of that is a transfer of electrons. All of that is a reduction or oxidation reaction. Okay. So we have to be very careful with that, and you can definitely fall down a rabbit hole in looking at all of those reduction potentials, and up to you if you want to look at it in detail. I actually can't remember if I left that question in the homework or not. I may have decided to delete it.
Which question?
It's a question in chapter 20 about ranking the different steps of your electron transport chain based on the reduction potentials.
No, you left that one out, but there is one.
I did
that's
pull that one
sorting-
out.
There's a sorting one for the complexes.
So there's definitely a couple sorting ones. There was only one,
Based on
but
reduction potential, I don't think.
there's not one based on, okay, and then I may have pulled it.
Just sorting.
I don't know, I think it is.
Yeah, because
Yeah.
you had to rank it from fresh to, but it's easy
Yeah.
once you, it's easy because it starts at the.
Yeah,
it starts
you
at
just
the
follow,
lowest and the
yeah.
highest,
You're
just-
So they've seen the homework, so that's a little bit unfair. What we're talking about here is looking at the reduction potentials of these. This is about as far into it as I want to get with this. What's happening with those reduction potentials is we're looking at our electron carriers. NADH is an electron carrier because it has those electrons. Coenzyme Q, once it picks up H minus, is an electron carrier. Okay? So where does the transfer occur? Does it go from NADH to coenzyme Q, or does it go from officially coenzyme Q H2, right? Yeah, coenzyme Q H2 to NAD+. Okay? Which way actually works? Well, we've already established that NADH is transferring electrons in, so coenzyme Q must be accepting those. Okay? One of the questions that an inquisitive mind might ask is, why is it doing that? Can it do the reverse? It turns out it can't do the reverse. So this is where you would go back to 152 stuff if you really wanted to, and you can look at your reduction potentials, verifying that you have a spontaneous reaction or a non-spontaneous reaction.
And
Okay?
it can't overcome that energy.
And it won't reverse. So Gibbs free energy, I didn't want to deal with symbols last night because it changes my font around too much and it's irritating. So we're looking officially at our change in Gibbs free energy. Our change in Gibbs free energy needs to be negative. So that's the Gibbs free energy of our product minus the Gibbs free energy of our reactant, and that needs to be negative for a spontaneous reaction. When we look at cell potentials, anybody remember what the sign on a cell potential needs to be for a spontaneous reaction? It needs to be positive. they're ultimately still looking at the same thing. We're looking on, is the reaction spontaneous or not? Okay? Why do we have this difference between positive and negative? The explanation that I come up with for at least 152 is that these two were separate fields that then merged their concepts, and each field said, no, I'm not changing my understanding of it. Okay? So when you go through to look at the relationship between Gibbs free energy and your cell potential, it's G equals negative RT, the natural log of your cell potential, which means your cell potential is positive, your Gibbs free energy will be negative, all because of that mathematical symbol. Okay? I don't think there's any other mathematical reason on why those charges there are established that way, other than somebody said, I want it to be positive for cell potentials, I want it to be negative for Gibbs free energy. And that's coming from how they calculated and established those values. Sweetness, awesome, we just deal with it. Okay? So how could we go through and evaluate that NADH must shuttle electrons to coenzyme Q and not the reverse? So what we could go through and, whoa, sorry, my screen just freaked out. We could go through and look at where these things are. So, ooh, this is going to be hard. I'm going to try to underline this. So there's our NADH, NAD plus reaction. And where's our CoQ? There's a variety of CoQs here, but I want to make sure we're looking at... it's actually not going to matter too much, but I'm going to underline the 2-electron one. Okay. Could we potentially take our electrons from our CoQ in its reduced state and transfer those to NAD+? Okay. Well, the reduction potential they've given here is for reduction, not the oxidation process of it. So if we're going to look at the oxidation, what do we have to do? We flip the equation, which means what happens to the sign? It goes negative. We would then add that to our NAD+, because it needs to accept those electrons. So we would just add that to here, and we get what for a cell potential? A negative number. Is a negative cell potential a spontaneous reaction? No. So that method doesn't work. So what happens if we then deal with looking at our NADH process? Okay, well, let's flip the NADH reaction, because what I'm trying to do is shuttle those electrons up to that guy. If we flip that reaction, what happens to the cell potential? It goes positive, and now I can add it directly to my CoQ, and my cell potential would be a positive value. That is a spontaneous reaction. Okay? So what we could theoretically do is, once we know we have all of these electron carriers, or what these electron carriers are, and their redox reactions, we can organize them according to looking at their steps through our reduction potentials. Okay? How important is that? Not all that important, but I at least wanted to address it. We've addressed it. That's about all I want to say with it. Do you guys have questions on that? Okay. I did delete a lot of questions on calculating delta G's and cell potentials, and that was just, uh-oh. Okay,
So you're not planning on putting something like that on the exam then?
No. Absolutely not. Okay, come on, reconnect. Okay, sorry, back with you guys. So, nice little pretty picture of everything that's happening here. We got complex 1, complex 3, complex 4. You'll notice that you don't see complex 2. Why not? No, it's still in the same membrane. Why do we not see complex 2? Remember, complex 1 and complex 2 both feed into coenzyme Q. If we look at this nice drawing, where would I draw complex 2? Because it's now a two-dimensional drawing, we can't draw complex 2 in there also feeding into it. Okay, so it's not very easy to see. If you think back to that first slide that we saw, you saw that our complex 2 was off to the side, feeding in. Okay? Just the way they've formatted this, you can't draw that in very neatly. Okay? But complex 2 is still feeding electrons into our coenzyme Q. So what ends up happening? Here's our intermembrane space. Here's the matrix. The matrix is on the very, very inside. Okay, so what happens is NADH comes in, shuttles its electrons into complex 2. This is where it gets fun. It hits FMN. What's FMN? Flavin mononucleotide, roughly the equivalent to FAD. Okay, it's for all intents and purposes the same thing, except you're missing that second nucleotide. Okay, that shuttles electrons to iron. Okay, or officially an iron sulfur complex. Okay, why might iron be a good thing to shuttle electrons to?
Positive.
Iron, being one of our metals, has multiple oxidation states. As a metal, we have very commonly +2 and +3 for iron. So iron is very willing to kind of shuttle those electrons back and forth. Once the electrons have been shuttled to our iron to make it a +2, okay, it now kind of wants to dump those electrons off somewhere else. So what it will do is end up changing its oxidation state. You'll notice this is kind of fun. To, again, FeS. Well, what changed? The oxidation state of the iron changed in that process. But as it changes...
When they show the charge.
Forest trees. Forest trees.
What happens to sulfur when it bonds with the electrons? Does that?
The sulfur is not picking up the electrons. The
Right.
iron's picking up the
But
electrons.
does it fall off, or does it?
It's going to kind of coordinate, and that is going to be a much more complex topic than you really
So
want to
you're
evaluate.
not going to ask us to like
What happens to the
explain
sulfur?
each of
No.
the mechanisms for the electron transport.
I'm not going to expect a mechanism at all for the electron transport chain. Okay. But what ends up happening is to change your oxidation state for your iron to that lower oxidation, did I say that right? Higher oxidation state, I think. We're going to shuttle H plus across the membrane. Okay. So it helps facilitate the transfer of H plus. Okay. And it's going to do that by likely changing the structure of coenzyme Q. So our iron shuttles its electrons to CoQ. In the process of doing that, it helps H plus get across the membrane. Okay. So that's your biggest signifier on this. NADH with electrons donates electrons into complex 1. Complex 1 does a little dance with the electrons. As it does that little dance to shuttling them out to coenzyme Q, it helps facilitate the transfer of H plus across the membrane. That's what you want out of that.
Does it transfer it into the inner membrane or just into the outside?
It transfers it into the intermembrane, okay? Because you're focusing on where this is happening. This is happening all on the inner membrane. Okay. It's in the inner membrane is where this is happening. And what you're doing is transferring hydrogens from your matrix, the very interior, to the intermembrane space between the inner membrane and the outer membrane. Okay. So I'm going to attempt to go, I may regret this. Yeah, I will go back to that in a second. Come on. What we're looking at, there's our complex 1. That's a 1. Okay. What it is doing, I don't know why my pen gets so twisted, is shuttling hydrogens from in here to out there. Okay. Which is why, as we do our electron transport chain, that intermembrane space has a very high concentration of H+, because you've taken all of the H+ from the matrix, the inside, and dumped them into that intermembrane space. That's what's generating that battery. Yes. Oh, you want to...
I have a question on that. Wait.
Ah.
The last one. So is it for complex 1, where it says NADH plus H, does that mean that it's NADH2? No?
I've really been hoping, so we keep coming back to what's going on with that extra H+. That extra H+ is generated when we oxidize your alcohol down to a carbonyl. That H+ comes from the hydrogen that's attached to the oxygen. It's separate from the NADH, which is why we don't show this as NADH2. That H+ is free floating from your
NADH
reaction.
isn't donating an electron to the transport.
NADH absolutely is. NADH goes from NADH to NAD+.
There's 2 electrons that go in.
It's 2 electrons from the hydride. Okay, so that's how you go from NADH to NAD+. Okay, you are transferring 2 electrons.
2 total, or just 2 from NADH?
2 from NADH.
And then 1 of just the hydrogen ions?
So you were transferring 2 electrons in from NADH and hydrogen ion from the solution.
Okay. Got
Right.
it.
You said you had another question back on the mitochondria drawing?
Yeah.
It's not the latest, it's actually way back.
The,
Come on.
If there's supposed to be a low concentration of hydrogen ions inside the membrane, then
Sorry,
how did they get there?
my screen is having problems. What is going on here? Oh, there it
That
is.
one.
So what ends up happening is in your Krebs cycle reaction, right, you're generating NADH. At the same time you generate NADH, you generate H+. That H+ is now getting transferred across.
Oh, okay. So it's always, yeah. And it
So
shuttles
you
it
are
back.
still constantly making that H+, and the electron transport chain is constantly grabbing that H+ and shuttling it across the other side. Do you guys have questions, or you guys self-resolve?
What makes it want to go for what initiates that transfer?
What initiates the
And then
transfer?
an enzyme
The,
back out of the.
okay, this recording, if anybody actually is listening to this at home, is going to have some problems. Because my computer and the iPad are not syncing up very well. Where are we? That's where we were, right? What's facilitating the transfer of the H+ across? Complex 1 does it. Complex 3 does it.
Oh, okay.
Right? Complex 4 does it. Right? Each of those complexes are shuttling H+ across. Once we've got coenzyme Q, we actually call it a coenzyme Q cycle, because what happens, we take coenzyme Q in a very small area, make it H2, it then dumps its electrons and hydrogens into complex 3. Well, now it's back to CoQ, so what does CoQ then do? Diffuses back through the membrane, back over to either complex 2 or complex 1, becomes coenzyme Q H2 again, shuttles back to complex 3. Right, so it's referred to as the Q cycle because it's constantly cycling between its oxidation states in a relatively small space within the membrane. Right. Once complex 3 has picked up the electrons, it goes through yet another electron transfer cycling process to help facilitate again the transfer of H+ across the membrane. You're going through a variety of another iron-sulfur cluster, you're going out cytochrome C. Cytochrome B is involved in the Q cycle. Right, that's how we're kind of getting the electrons into the cycle and facilitating transfer of our H+. Once our electrons make it out to cytochrome C can then diffuse across the top of the membrane. It's not inside the membrane now, it goes across the top to complex 4. As your electron transfer goes into cytochrome A. Cytochrome A now has the electrons, which can facilitate the transfer again of hydrogens across the membrane. At the same time, what it does is it reduces oxygen into water. So that whole oxidative process is really happening at that very end. Right? You might say, well, that's kind of weird. We're saying it's an oxidative process where ultimately what did we do to the oxygen? We reduced it. But to reduce the oxygen, what did you have to do? Oxidize everything else.
So did the chemists come up with the cytochrome starting with B, going to C, going to A, or is that a biology thing?
It's going to be a mix of both, because you need someone to be able to isolate those. Once somebody's got them isolated, then you can go through and process it. And so that discovery, the textbook actually mentions looking at different inhibitors. So what you can do is isolate this rough system and start dropping in inhibitors and see what happens. Does it shut down the process? Okay. Oh, it did shut down the process. What did that inhibitor target? And now start to use the inhibitor as a location beacon to then isolate that
It's just
particular molecule.
counterintuitive to start with B to C to A.
As far as your BCA process, what that's going to be is probably when they discovered it. So cytochrome A very likely was the first one discovered because A is the beginning of the alphabet. Then what did they discover? Cytochrome B, more than likely. Then they discovered cytochrome C, and then they discovered that, well, it turns out there's not just one version of cytochrome A, there's a couple other versions in those, so we'll call those now cytochrome A number, right, to kind of keep them all separated. Yeah, this I would say is, as a field, I wouldn't blame on biologists or chemists, I blame it on both of them. It's biochemists trying to come up with a standard system. Okay? It's such a young field is probably the argument that is a reasonable one to make. It's such a young field that they haven't actually spent the time to come up with a legitimate nomenclature system to organize it all. It's now also reached the point where there's enough egos involved that the likelihood that any of those are going to pick up a legitimate nomenclature system is beyond hope. Right? So we just kind of have to deal with the system as it is.
So all those electrons going through that whole cycle, just making water and being used to produce water?
Two things. Yes, we're producing water. So it's that oxygen reduction that's helping this process along. And the primary feature that we're concerned about is the shuttling of H+. That's the big one, H+.
Why do they have a complex too if FADH is made through the citric acid cycle and donates its electrons into the electron transport chain? Those complexes aren't one.
How does FAD shuttle electrons into the electron transport chain? Where does it shuttle those electrons
It has to
in?
be oxidized.
Where does it shuttle those electrons in? It shuttles those electrons in, not at the beginning of the electron transport chain, but halfway through it.
Through complex 2 that's not shown on
Yeah.
there. Okay,
So complex 2 is, crap, the name of that enzyme. The one with FAD. I don't know, I remember the name of the enzyme. It's the succinate to fumarate. That enzyme in the Krebs cycle is complex 2. They are identical. They are one and the same thing.
So do these complexes
This
work
succinate
respectively
CoA?
with one another, or no, they just kind of,
Sorry?
do these complexes work respectively with one another?
They have to work with each other, yeah. So if
Does it have to go through complex 1 first before complex 2?
complex 1 and complex 2 are independent of each other.
Okay.
Okay? Complex 1, they both feed electrons to CoQ. CoQ can then feed further on down the line. Okay? Complex 2 cannot feed electrons to cytochrome C. It can only feed them to CoQ.
That's so weird. In chapter 20, it's succinate oxidioreductase, but in the citric acid cycle, it's succinate dehydrogenase.
Because the succinate dehydrogenase converts it from succinate to fumarate, but then it's
Yeah.
this enzyme that takes it from FAD to.
Which is inextricably linked, yeah.
Okay.
So which one were you talking about? Each cycle you lose 10.
So those two are linked to each other in this process.
Okay.
Do you lose 10 hydrogen ions, or is it?
Wow, this is really freaking out. What's that?
Do you lose 10 hydrogen ions as a result of the electron transport?
Man, it goes too fast in one case, goes too slow in another. What do you mean lose 10 hydrogen ions?
That are being shuttled out?
10 hydrogen ions are shuttled across the membrane for this process.
So complex 2 is the same as complex 1 in terms of the hydrogen ions that it loses? Because it doesn't.
Complex 2 is not in this.
Right.
Complex 3 will shuttle 4 hydrogen ions across, just like complex 1. I take 1 NADH in, 10 hydrogens shuttle across. Okay. 4 from complex 1, 4 from complex 3, 2 from complex 4. That is all coming from a single source of NADH.
Seems like you're using complex 2 as the same instead of complex 1.
Complex 2 is not on this figure. You can't use complex 2. Okay? So once you go from complex 3 forward, yeah, it's going to be the same. But complex 2 is going to transfer something different than complex 1 because they're different enzymes. Okay? So they may end up being the same, but those are two separate topics.
So I have a question. Does that mean each NADH will produce 10?
Yes. It's a little bit more complex than
Yes,
that, but once you've got NADH starting at your electron transport chain, 10 hydrogens.
You're going to end up with 10 hydrogen.
Yeah. And it's based on the evidence that we have, there's less arguments about that than there are other topics, if that makes sense. Okay.
It's along the same lines as NADH produces 10 for 4 input. Or 4 outputs, excuse me, in the first complex.
In the first complex, it produces 4.
What does FAD complex do?
Is FAD discussed yet?
No, it's
Not
gonna
yet.
be an intermediate.
FAD is coming in through complex
okay.
2.
I get
We
you.
can't talk about FAD yet because it's not in here yet.
Got it.
That's coming from complex 2. So within complex 1, you're sort of bringing in FAD because what does NADH immediately transfer the electrons to? FMN, which is roughly equivalent to FAD. Okay? It's membrane or it's enzyme bound, hence the E. Okay? It is not the FAD coming from complex 2. Okay? It is an entirely different system. You're transferring those electrons into FMN. That goes through its old fun oxidative cycle, which is what they're trying to show now by breaking that image that we had earlier up into smaller pieces. So we go from NADH to our FMN. The FMN can then shuttle electrons to our iron cluster. The iron cluster can shuttle electrons to coenzyme Q. Ultimately, all that's happening is we are trying to push electrons to CoQ. Okay? That's all that happens. We could go through and look at the structure of that. We've talked about where you're putting on your electrons, and this is where you can get into the radical versus the other one, heterolytic cleavage. Since the radical kind of works nicely, let's just do that. Kind of ran out of space to our other hydrogen radical. Okay, so we are picking up 2 electrons and 2 hydrogens. How we decide to split those electrons across the hydrogens is going to come down to a mechanism. As long as we can reasonably agree on that, you're fine either way. So there's a radical version of it as we go through to form those bonds. What if we made it non-radical? Well, let's take H- and form a bond with that nitrogen. Then what's the problem? Nitrogen first has too many bonds. Shuttle the electrons. Shuttle the electrons, and that nitrogen would then be negatively charged, which could then react with H+. There's our last step. Okay. Exact same overall process, different mechanism behind it. How do we know which mechanism is the one actually running? That's where you'd have to pull these things out and look at how each of them react. Problem with pulling them out of the enzyme to see how they react, you've now changed their natural state. You're potentially not looking at what's actually happening within the enzyme. Does it matter? From a physical chemist's point of view, it absolutely matters because what we're trying to do is mimic that. Okay? This is an awesome process that does oxygen reduction. That's really cool. That generates power. So if we're going to try and make an artificial cell do this, we want to know how that's done specifically so that we can mimic it. From a biologist's point of view, absolutely don't care because it does it. I don't need to know it. I just need to know how I can stop it from happening. And to a certain extent, I don't need to know the specifics of how it happens to stop it. Okay. From just a general chemist's point of view, I can tell you mine. I like the heterolytic because that's easier to follow. There's a crap ton less arrows. It's easier for me to see. It's easier for me to manipulate, and I get the exact same result out of it. Okay? So everybody's going to have a slightly different spin on what they want to come out of this. The exact details of it aren't super important. Complex 2. Since everybody was excited about complex 2. Our complex 2 is coupling the succinate to fumarate. There's our complex, which generates the FADH2, which is enzyme bound. That's due to its size complication. It does not transport very well. So FADH2 can then shuttle that to an iron-sulfur complex. The iron-sulfur complex can shuttle those electrons to coenzyme Q.
So FADH2 is enzyme bound because of its size?
Yeah, that's going to be the easiest way to think about it. So where does this reaction occur? It's happening in the membrane. Okay? And have we seen this happen before? Absolutely. Where have we seen this happen before? It's the citric acid cycle. It's the Krebs cycle. Okay? So it's the same process that's happening. We're just using or getting the electrons from that oxidation over into our electron transport chain. Okay, that's what we're trying to do with this. Once we're at complex 3, okay, you're now taking the electrons in your electron transporter that came from enzyme 2 or complex 2 and complex 1 in CoQ. CoQ then goes through and starts doing a fancy electron transfer process, okay, through complex 3. Complex 3 is going to facilitate regenerating coenzyme Q so that it can be reused to pick up electrons from enzyme complex 1 and complex 2. And it's also going to be involved in shuttling those electrons all the way out to cytochrome C. Okay? When you go through and look at this, coenzyme Q, I thought about talking about this in great detail on what's happening with coenzyme Q. And based on time, I'm going to say it's not all that important for me to address. But you could potentially go through and evaluate it. That's what they're trying to show on that far side. You'll notice that if you look at how they're at least representing the electrons in this process, what type of mechanism do you think we're running through? Heterolytic or radical? It's a radical process. Okay? It's accepting a single electron at a time. Okay? How do we know that? This is, again, we pulled it out of the system and we tried to test it and we saw that we were running with radicals. Okay? What's that?
Sorry, I was running with radicals, sounds like a t-shirt.
Is not?
Running with radicals sounds like a t-shirt. Like a
Oh, yeah.
slogan.
It's not a bad one. Maybe we should set that up. Be a chemistry logo.
What would the image be?
Well, you guys can put that.
Yeah. Huh. Well, we have to think about that. Let me borrow that from you. I'll credit you. So we're looking at that electron transfer process into coenzyme Q. One of the things that's really facilitating this is if we take a look at the end result there. What is special about that molecule?
Special about it?
Uh-huh. Aromatic ring. Okay. Next thing that we can kind of address here,
It's fully saturated.
that functional group up at the very top, I've got it circled in blue, versus this functional group. This is an enol, and this is a ketone. That keto-enol process is of critical importance. It keeps popping up. Okay. So we are officially looking at a keto-enol tautomerization in this process. For that to occur, we do need electrons to facilitate this. Okay. Kind of a neat process. Where are we? Finally, complex 4. I haven't been mentioning them, but they've got enzyme names associated with them. Some of these are actually kind of nice. Cytochrome C oxidase. Why? What did complex 3 dump electrons into? Cytochrome C. So what does complex 4 need to do? It needs to oxidize cytochrome C. Okay, so it needs to transfer those electrons from cytochrome C out to our oxygen to reduce the oxygen. Okay, so we actually have a name that sort of makes sense. Complex 4's got all of those extra cytochromes involved with it. Interesting new thing that pops up, just because I happen to notice it now. We're looking at copper now instead of iron. Okay, why? It very likely has to do with the reduction potentials for iron plus 2 to iron plus 3 versus copper 1 to copper plus 2. Okay? So the enzyme is built around transferring those electrons. The potential for iron plus 2 to iron plus 3 probably does not work in this case and would not facilitate the electron transfer according to our reduction potentials. So it needs to be a different metal, okay? Or it needs to be a different electron transfer agent.
Copper transfers an electron in that cycle, goes from 1 to 2.
Yeah.
Was that Cooper?
Depends which way you're, yeah, don't even bring in the Latin roots. You're looking at cupric versus cupris, yes. I never know the difference on that. I think the Latin roots are dumb. Nomenclature, right? Okay. But ultimately, again, all of this process, all of this electron transferring, and really all we're trying to do is show that NADH and FADH2 make a battery. They put positive charge on one side, not positive on the other. Osmosis is naturally going to want to occur where we balance that charge out. That's the electron transport chain. The whole goal, make a battery. Now that we have this battery, this high potential on one side and a low potential on the other, we want to take advantage of that process trying to even itself out. To do that, that's where we bring in kind of a nice circle that we've come through, because what did we start the very first beginning of the semester off with? ATP synthase. Guess what? Here's where it comes in. Okay? So ATP synthase is shown kind of crudely in this drawing, but it actually works pretty well. Okay? Our whole process through our electron transport was putting all the hydrogens in that inner membrane space. Okay? They want to balance out the charge, because we got a lot of positive on our inner membrane space. Okay, so it is very acidic here, and it's not so much on the other side. So as with all reactions, what we want to do is neutralize. To neutralize that positive charge, I need to transfer those back to the other side. I need to make it to a weaker acid. Sorry, that looks like a weird W, but it is a W. Okay. To do that, the hydrogens must move back across. This is where the mitochondria has built up a very rugged membrane. That inner membrane does not allow for the passage of hydrogen ions. Okay, which means we've got our dam. So what ends up happening is there are particular pores within that that can allow for the shuttling of hydrogen across. And hydrogen absolutely wants to jump across this membrane. Okay? But the only way it can do it is by going through a particular enzyme. That enzyme is ATP synthase. And so what ends up happening, okay, which I don't know if you guys remember much of that video, we have a hole on one side of the bottom of that membrane. If this just went straight through, we got nothing. Okay, so what ends up happening actually is that we have a hole on one side and a hole on the other side. The hydrogen must go in, come around, and out. That process of coming around causes the enzyme to change its structure. It must rotate. Well, as soon as we have rotation, we have energy. That energy gets translated up into the upper portion of our ATP synthase. That energy actually grabs ADP and phosphate, grabs them and brings them near each other. Once they are close enough to each other, they can react. We can produce ATP, and it can then release those out. Fascinating enzyme in its system. ATP synthase is coupled to our osmotic neutralization. So you may hear coupling, and this is a question in the homework too. The electron transport chain does not produce ATP. It produces a proton gradient. A secondary enzyme takes advantage of that proton gradient to produce ATP. Okay? So even though we call it oxidative phosphorylation, the phosphorylation process itself does no oxidation, does no reduction. Okay? Electron transport does our oxidation reduction to give us a very high energy state. ATP synthase is now the generator within that dam, producing the energy. Okay? So if we're going to put in a drug to slow down that energy production, we've got a variety of ways we can target it. Okay? One option is targeting how ADP and ATP interact with that outer surface. Okay? If we can prevent them binding, that's not going to change any of this process except for the official binding of the ATP. We will still get hydrogen flow coming back through the bottom of this. Okay? But I can't translate that into ATP, and I've lost connection again. I can't get ATP because I've prevented it from connecting at the top. Okay? Or I can block something by preventing the hydrogen flow from coming back across. I can mess with that bottom portion. Or I can mess with the electron transport chain so that I don't ever form the proton gradient. Okay? So we can use a variety of drugs to target different aspects within that entire process, okay? Which doesn't necessarily shut down any one of those things, but it can slow the process. Okay? Or it can put it to the point where both things can't happen.
So is ATP generated in the matrix? I thought it was once it gets...
ATP is generated in the matrix, yes.
I thought it was going to shuttle it out and then it was generated like outside of the
Eventually that has to happen. Because we now have all of our energy, that's awesome, but we have it all inside the cell.
Right.
There's lots of processes that need that energy. So eventually what we're going to have to do is shuttle that ATP out of the mitochondria. Okay? Which, yes, that still needs to happen. Okay. How that happens is going to be yet another metabolic process that we could potentially look at if we wanted to, but at this point we're trying to just focus on the details here. Yes?
H plus stays in the mitochondria through this whole entire process. Never actually leaves. It goes in the
This membrane, the inner membrane, is
Impermeable.
that word, to hydrogen ions. The outer membrane,
Impermeable.
turns out not. Hydrogen ions can pass freely across that one. Okay. Which does seem a bit odd because
That's just counterintuitive, because I would think then you would have a change in pH outside the cell, or
you
or
could
outside
potentially
the
see that
mitochondria
acid
within the cell, and
contrast,
not
yeah,
within the
changing the
cell.
pH outside. So in theory, this process is happening fast enough that we don't worry about too much diffusion of that H plus out of the cell. Yeah, I thought that was odd too. Okay. But that's what's happening. Our ATP synthase, we can look at a closer image of it. This is where your hydrogens are getting shuttled around. Okay, there's the circle that we were talking about at the bottom. Our hydrogen comes in. It actually officially comes in on this side, and it comes out. I know I can't draw this very well. It comes out on the back side. So our hydrogen comes in, it rotates all the way around, and then comes out. That rotation is translated through the stator, which I believe is something referenced in actual engines, which causes a rotation up here, or causes enzyme configuration changes here. If this whole thing rotated, this part never changes. Okay? So what we've got is the green and the red and your C subunits down here all rotate. The top part does not rotate. It's actually locked in place due to this arm. Okay? Which means as the interior rotates, it causes it causes protein structural changes to the top subunits, which is facilitating ATP coming in or out. Okay? So kind of a fun little unit. We can look at all sorts of diagrams of what's going on with it. If we had had time, I was going to look up or try and find a nice little movie of it, because they were actually able to attach a gold nanowire to one of these, to the stator, and then pumped in a bunch of ATP. Well, what ATP is going to do is start to turn into ADP through the enzyme and cause the stator to rotate. How do we know it's actually rotating? Well, if there's a gold wire on the end of it, what happens as that stator rotates? The gold wire rotates. Okay? So we have actually proven that we do have physical motion within this. Okay? So it's a really neat process. A bunch of different states come out of this: open, loose binding, and tight binding. There is a question in the sapling that is looking at this figure. Address what's changing at each process, where things move in and out of it. I think you should be able to figure out the question. It is kind of challenging, but I don't think it's too bad. Okay? It's just a question of interpreting the figure. Oh, and I was supposed to delete this slide because it's already on that previous one. PO ratio, your textbook mentions it, might be kind of important. What we're talking about is phosphorylation to oxidation ratio. So if we look at the amount of phosphorylation occurs per oxidation, you'll notice that we get different ratios out of this, which is what a lot of you were starting to ask about. You referenced the H+. Okay? We're actually now looking at what happens when you transfer electrons from NADH. You actually produce more ATP than when you transfer electrons from
So maybe that's why it's more likely to
FADH2.
be more effective.
It could be more effective. We could look at a reasoning like that. Yeah, you're again getting into origins of life kind of arguments. Last thing, I know we're almost done, but this is pretty much the very end of it. Glycerus, not glycerus. The thing with glucose, glycolysis. Glycolysis is happening in the cytosol, okay, which is producing NADH and all of those fun little pieces. Those pieces don't transfer very well into the mitochondria. So we need to get that material into the mitochondria. To do that, we've got a couple different shuttle processes. Either your glycerol phosphate shuttle or your malate aspartate shuttle. Okay. Why is that important? Well, those are going to change how the energy, or how those high energy molecules are transferred into the mitochondria. Okay. If it's a more efficient process, what's going to happen to the amount of ATP produced? We should get more. If it's a less efficient process, we should get less. So depending on which shuttle mechanism we have, we're going to get varying amounts of ATP. Okay, so your book had some nice kind of figures just showing on how we're trying to get those molecules into our mitochondria, either through the glycerol phosphate or through your malate aspartate shuttle. They're just looking at structures and how you're transferring across that membrane. They're very particular molecules that can move across the membrane. Not all of them can do so. The big point comes out of this right here at the very end. If we follow along the glycerol phosphate shuttle, okay, this is kind of a nice slide because it's got all sorts of, or a nice table because it's got all sorts of information on it, but the big conclusion is right there at the bottom. You're looking at the official yield for ATP from beginning to end.
Pretty similar.
Okay, very, very similar, but not the same.
So which one is used more on?
Use more is a little bit of a hard question to answer. Which one produces more energy? That's a straightforward question.
Right, yeah.
Okay, the one that produces more energy is going to be your malate aspartate shuttle. Okay, that's going to get you 32. Okay, if you follow the glycerol phosphate shuttle, you get 30. Are these numbers debatable? They were at least 10 years ago when I first took the class because they addressed this issue in class. Said according to the MCAT, you only get 30 ATP molecules. Okay, so if you're asked the question on the MCAT, it's 30. Don't answer anything else. I know we said it's 32, but it's 30 on the MCAT. Depending on which mechanism you're looking at, you're going to get a slightly different amount of ATP. And as research has continued within, something just happened? Okay, I must just lost connection. Sorry. As we start to learn more information about it, we're going to refine further how much ATP is produced in each of those cycles.
So they use varying degrees of like NADH and FADH too?